In this video, we're going to be looking at cell cycle and the different parts of the cell cycle and what occurs in each cycle. So if we look at the entire cycle here, it is separated into a few phases. We have interphase, which is considered a non-replicating phase, and then we have what is over here. An interphase goes from G1S to G2. That's all considered to be part of interphase. And then we have over here the M phase, which is the mitotic phase, or the cell replication phase. All right. As we see in G1 right here, cells are going through their normal activities, normal metabolic roles, and different types of cells can have different lengths of time with which they spend in G1. A cell, like a skin cell, would probably have a relatively short G1 phase versus a neuron, which basically stays in G1 forever because they lack centrioles, which gives rise to the thought that neurons are amitotic, or they don't go through mitosis. So G1 stage is just the cell's doing what it's supposed to do. It's living its life. The S phase, right here, all right, is DNA replication, and we're going to look a little more intently about what DNA replication entails. This happens after G1 because the cell is getting ready to go through mitosis. So it needs to duplicate its DNA so that each of its two daughter cells can have the same amount and identical DNA. So if we look at DNA replication here, and so this is what is occurring in the S phase, DNA ends up becoming replicated, and this occurs inside the nucleus of the cell, and the end result of DNA replication is two identical strands of DNA, and they both have one strand of new DNA and one strand of old DNA. And if you remember when we talked about this in Chapter 3, this is called being semi-conservative. So the end result of the S phase, or DNA replication, is two strands of semi-conservative, yet identical DNA. So let's go back here to our cell cycle. So the cell has done what it's supposed to do in G1, it's gone through S phase, it's duplicated its DNA, and now it's going to be getting ready to go into the mitosis, or the M phase. But before it happens, there's one last growth phase in interphase, which is called G2. And what happens here is that the centrioles are going to be duplicating. And the centrioles are an organelle that are responsible for producing the mitotic spindle fibers that will be pulling apart the chromosomes and pulling them to polar opposite sides of what's going to become the two new daughter cells. All right, so let's look at M phase. So just to review, interphase has three phases. It includes G1, S, and G2. What is G1? Just cell doing what it normally would do, okay? S phase is DNA replication, which results in two semi-conservative strands of DNA. And then G2 is a final growth phase. It's a prep phase getting ready for mitosis, or the M phase. And in G2, that is where these centrioles are going to replicate. Now we're going to look at, mitosis, or the M phase. And what happens is that we have DNA that is in non-replicating form is chromatin, or chromatin. All right, it looks like spaghetti strings, and it has these histone proteins that are attached to them that just make it loose and uncoiled. When a cell is going to get ready for mitosis, what happens is that the DNA condenses and it coils, and it becomes visible structures, which we know as chromosomes. And so when a cell is getting ready to go through mitosis, let me get this out of the way here, we have chromosomes that are going to start becoming visible. So this loose DNA is going to condense and coil and become these chromosomes, which look like this. because the sister chromatids are attached to each other. Okay, so in prophase, what happens is that the chromosomes become visible. Okay, the nuclear envelope, all right, which is actually the outside of the nucleus, dissolves, and we have these centrioles that start migrating to the polar opposite sides of the cell. And as you can see, they're leaving behind these little mitotic spindle fibers. So that is step 1, which is prophase. Step 2 of mitosis, we have the chromosomes lined up along the middle of the cell, which is also called the metaphase plate. We have these centrioles, which exist in pairs that are at the polar opposite sides of the cell, and we have the mitotic spindle fibers that are going to be connecting to the center of the chromosomes. Step 3 occurs when the chromosomes start being split, and each sister chromatid separates, and the mitotic spindle fibers are pulling them towards the opposite ends of the cells. And so what happens is that half of this double genetic material is going over here, and the other half is going over here. Each new cell is also going to have a pair of centrioles. During the end of anaphase, we have this structure right here, which is called a cleavage furrow. That begins to develop a cleavage furrow. And this is splitting of the cytoplasm, which is called cytokinesis. Oops, must be an I-S. So that occurs in late anaphase. And telophase is actually prophase in reverse. So what occurs is that the, as you can see right here, the chromosomes uncoil and relax and become chromatin again. The nuclear envelope here starts to, it redevelops, okay? The cleavage furrow, these two cells will completely break into half, and cytokinesis will end, and we'll have 2 new daughter cells. They're not going to be as big as the existing cell, most likely, because the cytoplasm splits so that you can have part of the cytoplasm with some cytoplasmic organelles going to one daughter cell and the other half going to the other daughter cell. But telophase ends when cytokinesis is over. And that is the cell cycle.